next speaker is obviously Myron. And Myron is a professor of immunology at Queen's University. He said this morning he wake up very, very early to make the finishing touches on this talk. So looking forward to hear your talk, Myron.
So basically what I want to talk about is this whole signaling paradigm that we discovered over the years. And what we found is that a lot of these growth factor receptors, the receptor tyrosine kinases, for example, the EGFR, the TRK receptor for nerve growth factor, the insulin, there are about 20 of them that are highly glycosylated receptors. And the glycosylation have terminal sialic acids, whether alpha-2,3 sialic acid or alpha-2,6. And so basically one of the features with regard to the activation of the receptors is that they have to dimerize. And so basically what keeps them apart is this sialic acid, which causes a steric hindrance. And what needs to be done is to remove the sialic acid in order to dimerize the receptor, and you get downstream signaling. What we found here was that complex to the ectodomain of the receptor is this trimeric complex of neuraminidase 1, which is a mammalian neuraminidase 1, protective protein cathepsin A, and the elastin binding protein. The elastin binding protein keeps NEU1 from being active. So basically, one of the other features we found is that the matrix metalloprotease 9 is also complex to the receptor at the ectodomain, as well as the GPCR, G protein coupled receptor. And we found that as the neuraminidin B GPCR, the ligand for that is bumbasin-like peptide. So basically, what we have found here is that NEU1 matrix metalloprotease 9 and GPCR forms a complex, as you see here in at the cell membrane, complex to the receptor. We also found that the toll-like receptors on the innate immune cells, for example, TLR4, as well as the intracellular toll-like receptors 7 and 9, have an identical signaling paradigm that you see here that regulates the toll-like receptors, because both of these have to be dimerized in order to signal downstream. So basically, one of the mechanisms here is that when ligand binds to the receptor, there's a conformational change of the receptor that activates the GPCR signaling. Now, GPCR signaling activates MMP9, which is already complex to the receptor. The activation of MMP9 here removes the elastin binding protein to activate neuraminidase 1. Now, cathepsin A doesn't have to be active, and as long as NEU1 is complex to the PPCA, it becomes active. And once it becomes active here, it cleaves the alpha-2,3 sialic acids, that are terminal sialic acids on the receptor, removes the steric hindrance, and you get the dimerization receptor and downstream signaling. So one of the features here is that we've looked at a variety of different inhibitors of neuraminidase. We've looked at zanamivir, some of the influenza inhibitors, and we found that osmivir phosphate, which is the prodrug of Tamiflu, has a specific activity in terms of inhibiting neuraminidase 1 more so than some of the other ones. So basically what we try to do here is to validate the therapeutic efficacy of osmivir phosphate, or I can call it short OP, either alone or in combination with standard chemotherapeutics. And we've looked at pancreatic cancer, the breast cancer, prostate, ovarian cancers. What I'm going to do today is talk a little bit more about the breast cancer, especially the triple-negative breast cancer. So one of the features here is that if we shut down the neuraminidase 1, can we control the tumor growth? We can block metastatic spread, tumor neovascularization, the chemoresistance, as well as get long-term survival. And in one of the features here is that we use this intertopic xenograph mouse model called a RAG common gamma double mutant mice. And I'll talk a little bit about that mouse model later. So how do I go forward? Okay, too far. Go back, please. There, go back there. I can't control it here, Praveen. Praveen, next. Thank you. So basically, one of the features here, and we have looked at a variety of different aspects of the signaling paradigm in terms of looking at the therapeutic aspects of targeting neuraminidase 1 in multi-stage tumorigenesis. And we've looked at, for example, the tumor-mediated tumorigenesis with regard to the toll-like receptors, which is part of the innate immune system. Producing, for example, the pro-inflammatory cytokines that may be promoting tumorigenesis. We looked at tumor growth factor receptors, for example, the EGFR, TRAC, insulin, and several others of the tyrosine kinase receptors. We also found that the snail, as well as the CD31, which is the adhesion cell, endothelial cell involved in antigen blood vessel formation, feeding the tumor. This particular signaling paradigm seems to regulate the angiogenesis, as well as metastasis, and as well as chemoresistance. So basically, one of the features here is that we're able to shut down neuraminidase 1, and we can block all these different features of multi-stage tumorigenesis. One of the things I wanted to mention here that we did biochemical analysis in terms of co-localization, co-immunoprecipitation, lentivirus knockdowns, as well as animal models of deficiency for MMP9 and U1, and showed that it has that we can elucidate this particular signaling platform that regulates all these different receptors. Next, please. Too far. There it is. Awesome. So we published one paper here with regard to the triple negative breast cancer, and we were interested to see whether this osmolar phosphate monotherapy is able to target this tumor neovascularization, tumor growth, as well as metastatic spread in this mouse model of a human triple negative breast cancer. And these are some of the students involved with this particular study in collaboration with Ron Neufeld, who's a chemical engineer professor that collaborated with me. So one of the features here is that, as you can see here, that the patients with triple negative breast cancer have a typical high grading, more frequent relapses, and exhibit poor outcomes as compared to some of the other subtypes of cancers. And so basically, in the clinic, there are no targeted therapies that are very effective against triple negative breast cancer. Next, please. We looked at this advanced metastatic breast cancer, and as you can see, there's local therapy as well as systemic therapy. Local therapy can involve surgery, radiation, or when you look at the systemic therapy, there's different facets here in terms of whether these cancer cells express the estrogen receptor or the progesterone receptor. And there's, for example, the hormone therapy. And if it expresses the HER2, then you go through chemo as well as the antibody to the estrogen receptor. But the problem here is that if you have a triple negative, then the end result is to use chemotherapy. One of the features about chemotherapy is that the cancer cell becomes resistant and develops into a metastatic phenotype and disease. This particular feature of this article here is published in a chapter in this book by breast cancer, metastatic, and drug resistance, and it's available for reading. I've also want to acknowledge that we published a book on current application of overcome resistance to targeted therapies, and Serene, Cecile, Sabri, and Makish has contributed chapters to this book, which is quite interesting in itself. Next, please. So basically, one of the things what we wanted to do at the initial stages of all these studies is to see whether the neuraminidase, as you see here, plays a role in being activated. So this is having like a bird's eye view on the landscape of this signaling paradigm. Click next, please. So basically what we do is we plate the live cells on a circular glass plate. Then we can add these circular glass plate on a mounting media with forminana, which is a fluorogenic sialidase specific substrate. And when the neuraminidase is active, this substrate that's surrounding the live cells here becomes cleaved into this particular methylbelliferone, and at the wavelength of 450, it fluoresces a blue color. So basically in the control, you can see here that these cells do intake some of the forminana, and there's some activity within the cell with regard to sialidase activity, but surrounding the cells, it's clear. Next. And then when we add the ligand, for example, if we're looking at the some of these receptors, for example, EGFR, we add EGFR or even the insulin, whatever, we get this brilliant blue color. And this is indicated that that when the ligand binds to the receptor, it activates neuraminidase. And since there's substrate surrounding the cell, live cell, the neuraminidase cleaves the substrate, giving this blue fluorescent color. Next, please. And then when we have an inhibitor, for example, if we inhibit, for example, MMP9, or if we inhibit GPCR with specific GPCR inhibitors, or even using inhibitor to neuraminidase like osmolar phosphate, we can shut down that sialidase activity. Next, please. So we looked at triple negative breast cancer in terms of neuraminidase activity associated with EGR stimulation. And as you can see here, that when we add EGFR, we get this blue fluorescent image here with sialidase activity as a result of sialidase activity. We can block it with osmolar phosphate, and we can block it with anti-Neu1 antibody or with an MMP9 specific inhibitor. And so basically this sort of gives us an indication of that these different molecules here involved in the regulation of the neuraminidase activity. We also looked at some of the tamoxifen resistant cell line of the triple negative breast cancer. And we can see here that it takes a much larger dose of osmolar phosphate in order to block the sialidase activity. And that was quite interesting in itself. Next, please. Next, please. So we also looked at different dose response of osmolar phosphate in looking at the MCF7 cell line, which has expressing the HER2 MCF7. And we've looked at the 5 micromolar tamoxifen resistant cell line, as well as the much higher dose of tamoxifen resistant cell line. These particular cell lines were established growing in this particular tamoxifen for over a year, so they're highly resistant to the tamoxifen. And as you can see here, we're able to block this sialidase activity in these live MCF7 cells. Next, please. So we looked at also the cell viability of the MCF7, whether the parental as well as the tamoxifen resistant cell lines, and adding increasing dose of osmolar phosphate, we can reduce the cell viability in a very significant way. This is true for the MCF7 as well as the triple negative MB231s. Next, please. So one of the features with regard to triple negative breast cancer is this phenomena called the triple negative paradox, where the therapy for treating the triple negative breast is using chemotherapy, standard chemotherapeutics. And one of the end results is that you get a recurrence, relapse, and whether it's in the local systemic regions. And so basically you can have a good sensitivity, but bad prognosis, which is referred to what we call the triple negative paradox for as a treatment for this cancer. So one of the things we were interested in here is that if we use a very little dose of the chemo, for example, cisplatin at 1 micromolar, as well as the 5-FU, Paxil, cisplatin, gemcitabine, as well as tamoxifen, but increasing dose of the osmolar phosphate, you can see here that there was a significant decrease with increasing dose of osmolar phosphate in order to cause a decrease in cell viability. And that was quite encouraging that what we're seeing here is seem to have an effective aspect of treatment. Next, please. So basically, it's very nice to do tissue culture and showing the effect, which was quite promising. But the end result is to see whether we are able to reduce the tumor growth in vivo mouse model of cancer. We use this particular mouse model. This is the RAG common gamma mouse model. And one of the features here is that it has a stable phenotype characterized by an absence of T lymphocytes, B lymphocytes, but one of the important features here is that they lack NK cells. And NK cells is one of the features about NK cells is that if you try to look at the metastatic phenotype of the cancer, the NK cells, if they're prominent in the mouse model, it can prevent the metastatic spread. So this was a very nice mouse model in order to show the effect of our drug treatment in preventing metastatic disease. Next slide. This gives you, this particular mouse model has been published, but one of the things that's important to highlight here is that it does have an innate immune system. It has granulocytes, monocytes, macrophages, dendritic cells. It's all functional. So one of the features here is that the innate immune cell does play a critical role in tumor genesis in terms of the tumor microenvironment. Next slide, please. So we looked at, for example, in the RAG common gamma double mutant mouse model in terms of the triple negative breast cancer, the MB231. We did an implant with subcutaneous, and we found that the untreated, you see a tremendous tumor growth. But one of the features, what we found here is that the skin developed lesions. And when we looked at the necropsy here, the tumor developed a very bloody vasculature of the tumor, which was quite interesting to see. But when we looked at, for example, when we treated these animals on a daily dose with 30 mg per kg of OP, we can see a difference here in terms of tumor growth as well as tumor vascularization. And here you can see that with the 30 mg per kg OP, we're able to get a growth pattern. Some mice had a much larger growth pattern. we've looked at for example, we're interested to see whether this blood vessel here, the bloody vasculature, and when we didn't see it here with this treatment, whether the CD31, which is the platelet endothelial cell adhesion molecule, which plays the cell-cell adhesion in terms of developing new blood vessels, we can actually see here that with the treatment here, we can reduce the CD31 with the treated tumor, but also we were able to show within the tumor that we can reduce the N-cadherin, increase the E-cadherin. And as you know, the N-cadherin is one of the features that establish the tumor become metastatic. And when we looked at the untreated here, there was a high level of N-cadherin and low levels of E-cadherin. Next slide, please. Well, we were really amazed by these results here, because when we increased the dose of osmolar phosphate to 50 mg per kg, one of the things I want to mention here is that we did a preliminary study looking at the toxicity of OP in terms of increasing dose, and we found that there is no toxicity of injecting this osmolar phosphate in normal mice. So that was a key feature here. But what we see here is that when we start giving a daily dose of osmolar phosphate, we can see here that it really controlled the tumor growth. And that at about 121 days, we were able to see the osmolar phosphate being removed from the treatment, and we got a completely reduction in tumor growth. As you can see, you didn't see any tumor growth at all. Next slide, please. So one of the features here is that very nice, since we have this very nice mouse model that lacks NK cells, we were interested to see whether we're able to see metastatic disease, especially in the triple-negative breast cancer. They do develop lung metastasis. And when you look at for example, the untreated control, you see here considerable micro-metastatic clusters of the tumor seeding the lung. When we look at the 30 mg per kg OP, we see less of metastatic tumor in the lung. And when we looked at the 50, we didn't see any at all. And so basically, one of the features here where we're interested to see is how is this metastatic tumor within the lung here is able to form itself, and we were interested to see whether the sialic acid does play a role in this formation. Next slide, please. So basically, one of the features here with regard to metastatic disease is that when you have a primary tumor, in this particular mouse model, it was a subcutaneous cells injected. And then after time period, they started migrating, maybe through the tumor neovascularization, and then they go and see the distant organs. And so basically, one of the features here is that what we wanted to do is to see whether we can mimic the tumor microenvironment here that may be contributing to the development of this tumor metastatic nodule. And so basically, one of the features we're able to do here is that with the monolayer of triple-negative breast cancer, we added this cyclic RGD FK peptide, and after 4 days, these particular cancer cells, two-dimensional cancer cells, form a spheroid, which seems to mimic very similar to what we see in the lung metastasis. The next slide, please. Next, please. Next, please. Next. Okay. And so basically, one of the features with regard to the cyclic RGD FK This particular peptide was synthesized by my Russian collaborators in St. Petersburg and Moscow. And one of the features here is that this particular peptide here mimics the natural extracellular matrix proteins that may be involved in the cell aggregation. And this is probably due to the alpha 5 beta 1 integrins. And so as you can see here, the fibronectin here can have a direct effect on the integrin here. And so basically one of the features about the peptide here is that it's cyclic and it has these different features here. And one of the other factor here is we're able to conjugate this TPP, which forms a complex chemical bond with aspartic acid. And the next slide here gives you a little bit better representation. So basically, one of the features about the cyclic RGD peptides is that it's more rigid and it has lower entropy. It doesn't need a lot of energy for it to react. It has increased target specificity in terms of specifically targeting the integrin. And it has, and it's because it's cyclic, it's less susceptible to degradation by the proteases. And one of the features with regard to triphenylphosphonamide, the TPP, it forms this covalent bond with aspartic acid, but it also enhances the electrostatic interaction of the peptide with the target cell, which is the integrin. Next slide, please. So basically, involved in the formation of spheroid is that the TPP here, as you can see here, binds to the integrin. And the integrin here, as you can see here, is this highly glycosylated molecule. And what happens here is that you get this cell aggregation forming. And the next step here, there's a delay period. And that delay period where you get the TPP peptide here binding to the integrin, it gets internalized. That internalization activates the upregulation of the E-cadherin. And so this E-cadherin, you get the cell aggregation where you get two E-cadherins coming together to form spheroid formation. Next slide, please. So we looked at this very carefully, and we found that one of the features here is that since E-cadherins is highly glycosylated receptor as well as the integrins, so we were interested to see whether the sialidation of these molecules does have a role to play in the formation of these spheroids using this cyclic RGT peptide-induced self-assembly. And so basically, one of the features here is that the specific route, you got to go forward, please. One of the features here is that there's the specific sialic glycan structures on the cell surface that play a role in the ability of the cancer cells form not only these spheroids, the avascular multicellular tumor, but also it can be involved in the in vivo xenograft tumor formation. Next slide, please. So basically, it's rather a fairly simple procedure here. And when we take the breast cancer MCF7's two-dimensional monolayer, all you do is add a certain concentration of the cyclic RGD peptide. You wait about 3 to 4 days, and what we find here is that you get these spheroid formation, which is makes a very simple way of studying these 3D multicellular tumor formation. Next slide. And so basically, one of the things we looked at here is the MCF7, which is tamoxifen resistant. What we found here is that this particular the cyclic peptide here platform seems to potentiate for cell clustering and compaction. And we found here is that when we looked at the tamoxifen resistant CF7s, they readily form spheroids that much more tightly and much more forming the spheroid compared to the parental cell line. And so basically, this was a quite revealing study here. Next, please. So we looked at to see whether the sialic acid plays a critical role here. And so one of the things we were able to do is use neuraminidase, exogenous neuraminidase, vibrio cholera, at different concentrations, and to see whether we are able to reduce the sialic acid on the cell surface of these cells. And we can see here when we use MEL2, which is a lectin specific for alpha-2,3 sialic acid, or SNA, which is specific for alpha-2,3 sialic acid, you can see here that they do express on the cell surface, the receptors express these sialic acids, and they can be picked up by these lectins. But when we do the treatment with neuraminidase, we can reduce this sialic acid compound. When we study this in the spheroid formation, we increase the dose of the neuraminidase, and we can reduce this spheroid formation. So basically, one of the features here is that the cell surface content of the sialoglycoproteins play a critical role in spheroid formation, but also it could play a critical role in the formation of tumor in vivo, as well as metastatic nodules. Next slide, please. And so basically, the other thing we did here is we were able to treat the cells in combination with the cyclic peptide with MEL2, which binds to the alpha-2,3 sialic acid, as well as use SNA, which is against the alpha-2,6, and we were able to reduce the formation of the tumor. So basically, this tells us that the sialic acids do play a very fundamental role in spheroid formation. Next slide, please. And so basically, we looked at the spheroids that are made by the MCF7 using the cyclic INP peptide, and we can see here that they do express the alpha-2,3 sialic acid when they're formation, as you can see here. So one of the conclusions here with regard to this particular study here is that sialic acids does play a very fundamental role in cancer aggregation and compaction to form not only the spheroids, but also in vivo tumor formation, as well as when the cells migrate to distant organs. They use the same process here in forming metastatic nodules distant to the primary tumor. Next slide, please. So one of the things I wanted to mention here is that this is very nice because we were able to shut down, I haven't shown you all this data, but when we shut down the neuraminidase, we can shut down all these different features of multi-stage tumorigenesis. And so basically one of the things we were interested in to see if we can have a specific targeting of neuraminidase with particles. And next slide, please. So one of the things in collaboration with Ron Neufeld, which is chemical engineering, and with some of the students that were involved in this project, is that we were able to design this polylactic glycolic acid cylinder, where we're able to make this particular polymer where we can put osmolar phosphate crystals in there and make a cylinder. And so basically we were able to grow tumors in the mice, and then we did a surgical implant of this PLGA osmolar phosphate, which is did a surgical implant close to the tumor. And so basically one of the features here we found here is that as you can see in this diagram here, is that this PLGOP here close to the tumor here really reduced the tumor size, but also reduced the bloody vasculature of the tumor compared to what we see here in the untreated. One of the features with regard to this PLGA OP is that it had a slow release of the OP, which is the biodegradable features of the PLGA. One of the, so when we measured the tumor volumes here, in the red here is the PLGA OP at 20 mg per kg of OP. And you can see here, it really shut down the tumor growth compared to the untreated. We use a control here, which is the PLGA, which is empty without the osmolar phosphate. And you can see here that the tumor started to grow and then it decreased. And we said, wow, something's going on here. What's happening here? And one of the features here is that what we found here is that the empty PLGA seemed to have the blood vessels sequestering to the PLGA and removing the blood vessels from feeding the tumor. And this is probably one of the features what we're seeing here. Next slide, please. And we looked at, for example, the metastatic disease. And as you can see here, we were able to show that when we looked at number of metastatic clusters within the liver here, the PLGA OP here, we didn't see any metastasis in these treated and in these animals. Whereas the untreated here, they had huge metastatic clusters and nodules, as you can see here. With the empty one here, we were able to reduce the metastatic cluster in the liver. And this was true for the liver. So this particular cancer here metastasized not only to the liver, but also to the lung. And this particular treatment here, we're able to show that it has an effective of reducing that metastatic spread. And probably one of the features here is that the OP not only has an effect on reducing the tumor growth, but also it can reduce the tumor neovascularization. One of the features, one of the questions we asked here, maybe the PLGA itself may be toxic to the tumor. And we did a study here showing here that when we add the blank PLGA to the culture cells, it had no toxicity effect. Next slide, please. So that was a very interesting study. And we continued on doing some additional studies with my chemical engineer here, Ron Neufeld, with the students illustrated here. And we were able to look at where we were able to conjugate osmolar phosphate to polymeric micelle using this RAF living radical polymerization, which is a new active target treatment. So one of the things we were interested in here is that if we can make this micelle that has the inner core here, which is hydrophobic, and then the outside here is hydrophilic, where it has the osmolar phosphate. We were interested to see whether we can use the particular micelle to target the osmolar, the neuraminidase 1, and block its activity. One of the features with regard to this is that not only the micelle is able to target the neuraminidase 1, which is already complex to the receptor, but we got receptor endocytosis. So this is quite interesting here in that we were able to deliver a hydrophobic drug through using this micelle, targeting the neuraminidase, and it becomes receptor endocytosed. And then once it's inside the cell, it explodes and kills the target tumor cell. So we were able to do that. I think this is my last slide. So one of the conclusions here is that we believe here that this mammalian neuraminidase 1 is identified as a major target in multi-stage tumorigenesis. I'm saying that is that if we can reduce the tumor volume by considerably, we can reduce chemoresistance, we can reduce metastatic disease, and as well as the tumor neovascularization. One of the things I didn't mention here is that some of the later studies here we've done, we used this osmotic pump where it was delivering the drug osmotic phosphate on a continuous basis on an hourly for that dose per day for 42 days. And we were able to reduce the tumor volume by almost 95%, reduced metastatic disease as well as chemo resistance. One of the important things about this is that this constant delivery of the osmotic phosphate shutting down this neuraminidase is that the cancer has a compensatory reaction to react to different stimuli, and we need to be continuously perfused in order to prevent that from happening. And I'll stop there and answer.